of the scat and preserve it in DNA RNA shield, and then that's kept frozen until once I have all of my subsamples, I'll be extracting the DNA and then using the 18S rRNA gene and a mammal blocking primer to amplify the prey DNA. And then the species will be identified through DNA sequencing. So I have some preliminary results from Shannon Hogan, her master's thesis. So she was just looking at microplastics in gray seal scat. So she used 19 different samples from July 2019, November 2019, and January 2020. So in all of her samples, she found microfiber particles, and then in 95% of the samples, just microfragment particles. And then she also looked at the difference between the months. So microfiber particles were significantly different between months. So January 2020 had the highest concentration per weight of the scat, and then November 2019 had the lowest concentration of microfiber particles. Just the microfragment particles were not significant between dates. And then down here, this figure shows the kind of variation of colors that we found of the microfiber particles. So in July 2019 and January 2020, clear fibers were most prevalent, and then in November 2019, blue fibers were most prevalent. And then these are just pictures of what you might see when you're looking at a fiber. So these two on the ends are fragments, and in the middle we have a fiber. So when I include my diet analysis, I have some hypotheses for that. So my first hypothesis is that seals that consume a larger number of gatted species will have consumed a greater number of microplastics, and that's because two different studies found a significant positive correlation between gatted consumption and microplastic presence in Northeast Atlantic gray seals. My second hypothesis is that microplastic abundance will be highest in scat collected in winter months. That's because a previous study found that cod species made up the greatest percentage of gray seal diet in the winter, and cods are a gatted species. So combining those two, that's why I think microplastics will be highest in the winter months. And finally, I just have the significance of my research. So gray seals are a sentinel species to the ocean, so they can be studied to determine the broader impacts of stressors on the ecosystem. And then the diversity or importance of prey species has been underestimated when just using prey hard parts to analyze diet, so that's why I'm including the prey DNA part of my methods too, to see the difference between just using prey hard parts and using DNA as well. And then since gray seals do consume some commercial fish species, this research can also aid the understanding of human microplastic consumption. And then finally, this will be the first study to examine the relationship between diet and microplastics in gray seals using DNA metabarcoding, as well as the first to compare hard parts, metabarcoding, and microplastic analysis. So thank you for listening.